the BLAST alignment tool that will be used in Lab 4. So I'll just highlight a few options that will be investigated in Lab 4. Now, when you get to BLAST, the very first information that needs to be provided is the sequence to be used for the alignment. You could either copy paste a sequence, or the second option would be to retrieve a file with a sequence saved under the FASTA format. It's really important that files are under the FASTA format to make sure that BLAST can read the sequence and do the alignment. So this is the submission section. The other section is about the search set. So which database will you use for the alignment of your query sequence? You could leave it as the default option. It's a nucleotide alignment, so this is the nucleotide collection, or you could refine the search and you could say, for example, I want to do a search only within the human genome or the mouse genome, or there are also a couple of others options. For us, we'll just leave it as the default option, so the reference nucleotide collection. Then the program selection, we'll just leave it as is. And another thing that we will investigate in Lab 4 is the scoring, the parameters for the BLAST scoring function. And more specifically, these match and mismatch score. So by default, every match will count as a +1, a sort of bonus, and the penalty of -2 will be assessed for any mismatch within a given alignment. But these values could be changed, and there's an exercise asking you to modify the match and mismatch scores, and if you change those match and mismatch values, you'll definitely affect the alignment results. But by default, it's 1 for a match and -2 for a mismatch. A gap cost, a linear option means that the creation of a gap, a gap of 2 nucleotides will be associated with a penalty twice the penalty for a gap of 1. So this is a linear penalty. It's linear with the length of the gap. But there are other options, like the second one, the existence or the creation of a gap. It's a penalty of 5. And once a gap has been created, if there's an extension of 1 nucleotide, then it's a penalty of 2. So it's not a linear function. There's a higher penalty for the formation of a gap, and then a lower penalty for each nucleotide, extra nucleotide within a gap. So you'll have the opportunity to explore these parameters in one exercise of Lab 4. And then if you're ready to do BLAST, you simply press BLAST, and the